In 2019, the world was hit by a pandemic that changed everything we knew about health and disease. It was caused by a virus that attacked our respiratory system, making it difficult for us to breathe. But what caused this virus to be so deadly?
We now know that this virus, SARS-CoV-2, or more commonly known as COVID-19, gains entry into our cells through a special protein called spike protein, which is present on the surface of the virus. The spike protein binds to a specific receptor called ACE2, which is found on the surface of certain cells in our body. But how does this process happen? How can we stop it? Spike protein of COVID-19 plays a crucial role in the virus entry into the host cells. This protein binds to the ACE2 receptor, which is present particularly in cells found in the respiratory tract. The binding of the spike protein to the ACE2 receptors triggers a structural change in the protein, also known as a conformational change, which enables it to fuse with the cell membrane and enter the cell. These specific conformation changes in ACE2 that are triggered by the binding of spike protein have been extensively studied. ACE2 is a type 1 transmembrane protein, which consists of a large extracellular domain and a small cytoplasmic tail. The extracellular domain of ACE2 contains a region called the catalytic domain, which is responsible for the enzyme activity of ACE2. The spike protein COVID-19 interacts with the catalytic domain of ACE2, which triggers a conformational change in both the spike protein and ACE2. The interaction of the spike protein with ACE2 leads to a displacement of a loop region in ACE2, which exposes a deep pocket that binds to the spike protein. This binding induces a further conformational change in the spike protein, which enables it to fuse with the cell membrane and enter the cell. While vaccines and other treatments are essential tools in fighting against COVID-19, basic hygiene measures remain crucial in preventing the spread of the virus. One effective way to destroy COVID-19 virus is through the use of soaps, detergents, and disinfectants.
You may have heard from groups like the Center for Disease Control or the World Health Organization that best practices in combating COVID-19 involve washing your hands regularly with soap and water and cleaning surfaces frequently with detergents and disinfectants. But how do these actually help combat the virus? Let's start with soaps and detergents. Soap and detergent molecules have two main areas, the water-loving or hydrophilic head and the water-hating or hydrophobic tail. These components of the molecules cause the formation of what are known as micelles when mixed with water. Micelles will form around the COVID-19 cells and begin to interact with their own membrane. Since the main components of the COVID-19 membrane are phospholipids, a structure incredibly similar to the soap or detergent molecules composing the micelle, the individual molecules from the micelle will begin to insert themselves into the COVID-19 membrane, disrupting it, eventually leading to cell death. How about disinfectants? While there are many classes of disinfectants, the main one we will be focusing on are alcohols, as they are the most prominent disinfectant and can be often found in items such as hand sanitizer. Alcohol-based disinfectants have one main mechanism of action: clumping. What alcohols do is cause proteins in the membrane to clump together, which disrupts the structure of the membrane of the virus. This disruption, as seen with soaps and detergents, causes the viral cell to eventually die. The main difference between soaps and disinfectants in regards to viral cell destruction is the fact that soaps are much more specific. When we use the term specific, we refer to the fact that it attacks a smaller variety of cells, where alcohol-based disinfectants will target any cell that it can find. This makes soaps much safer than alcohol-based disinfectants in the long term. Another way for us to combat COVID-19 is through vaccines. But what kind of information needs research for the development of a vaccine?
Structural information is a critical component in the development of vaccines and therapies for COVID-19. In the case of vaccines, researchers use structural information to identify key regions of the spike protein that are essential for the virus entry into the host cells. By targeting these regions with the vaccine, the immune system can be trained to recognize and neutralize the virus, preventing infection. Several papers provided describe the use of structural information to design vaccines against COVID-19. One study used an advanced imaging technology called cryo-electron microscopy to determine the structure of a spike protein when bound to the human ACE2 receptor, providing insights into the specific interactions that drive the virus entry into the host cell. Another study used another technology, X-ray crystallography, to determine the structure of the spike protein in complex with neutralizing antibodies, which can be used to develop therapies for COVID-19. In addition to vaccines, structural information can be used to develop therapies for COVID-19. The structure of viral protease, which is essential for the replication of the virus, has been determined using X-ray crystallography, providing a target for the development of antiviral drugs. Other studies have been using structural information to design small molecules that can bind to and inhibit the activity of the spike protein, preventing the virus from entering host cells. Therefore, by understanding the specific molecular mechanisms involving the virus entry and replication, researchers can continue to develop effective strategies to stop the spread of this deadly disease.
Today, we talked a lot about how COVID-19 infects human cells, the mechanisms of some of the methods of combating COVID-19, and some of the research that goes into developing a vaccine. But why should you care? In terms of the average person, you may think, I'm not researching this disease. I shouldn't care about any of this information. But you could not be more wrong. First, understanding the mechanism of action can help you and many others understand that the infection is incredibly invasive to your body, with the virus having a great effect on the cells that make up your body. While you do not need to understand the intricacies, you do need to understand that the virus's mechanism of action is incredibly invasive and effective, which contributes to its highly contagious nature. Second, understanding the mechanism in which the virus can be destroyed by soaps, detergents, and disinfectants helps to illustrate the importance of these weapons against COVID-19 and other viruses. While washing your hands or using hand sanitizer somewhat frequently sounds like it may not help, it can actually reduce your risk of contracting symptoms drastically. Washing your hands alone is a simple but effective way to kill the virus and keep yourself healthy. But why would you want to know what data we need? Finally, understanding the data that needs to be collected and the research that targets that data helps to put into perspective what a concerted effort developing a vaccine can be. There's a lot of research that goes into the development of the vaccine, and while we talked about a small portion of that information, it is still important to have a general idea for what is looked for under the development of a vaccine. It takes a lot of time and money to develop a vaccine, and the fact that the COVID-19 vaccine was developed so quickly shows the power of our species when we focus all of our attention on one issue. Hopefully, it doesn't come to that again, but if it does, I have confidence in our capabilities.